All right, folks, I thought I would do a little video here on Chapter 3, maybe split this one up and do a Chapter 3 and a Chapter 4. So just a quick kind of run through and some highlights of Chapter 3. So Chapter 3 is cellular level of organization. So we're going to talk about cell pieces and parts and how they're kind of put together. And then Chapter 4 is tissue, so one kind of builds on the other. So in previous chapter when we were talking about biological molecules or organic molecules, the mention of lipids came up and triglycerides and diglycerides and things like that. The cell membrane is made mostly of these phospholipids, and so phospholipids are considered diglycerides because they have these two fatty acid tails. One of them may be unsaturated, so you get this kind of kink in the structure. But it has these two fatty acid tails with this phosphate group at the top, so phospholipids. That's where they get their name. You'll notice here that it mentions a hydrophobic tail and a hydrophilic head. Hydrophobic things are things that are afraid of water. Hydrophilic things are things that love water. And so what you end up with is... Right, so just reiterating, what you end up with is, maybe I should draw this out for you first before I make sure my pen is on. If I have a phospholipid that looks like this, and obviously I'm an excellent drawer, because this is in a cell, you're going to have water everywhere, right? And so if these heads are water-loving, right, they want to be attracted to the water, and if these tails are water-fearing, they want to turn away from it. So if you have multiple of these phospholipids together, what will happen are the tails will turn towards each other, and the heads will turn away, right? And so if I had another one here, the head would be here, tails, tails, head, right? So on and so forth until you go all the way around and you build a membrane. And they do this spontaneously. You don't need energy necessarily to put them together. And the other thing that'll happen is these, oops, let me erase that. Well, I hate this, but it does all right, I guess. The other thing that'll happen with these phospholipids is they can kind of move in this direction. They can move in this direction. They can move backwards. They can move forwards. This arrow is a little misguided though. They can't. What this phospholipid can't do is flip down here. If it flipped down, that would take energy. But they can move in all these directions back and forth, and it creates this kind of. It's called the fluid mosaic model. So the idea that this is not a stagnant environment, right? It can move around everywhere, and so what you end up with is something that looks like this. Right? Again, almost as good as my drawing, but not quite. So these phospholipid, the tails here are hydrophobic, so they turn towards each other, they exclude water, and then the heads are facing out, right? And so the water is against them. And so that just allows you to separate. So here I can have, I can have extracellular stuff and I can have intracellular stuff. I can have a barrier, right? And that's what membranes do, create a barrier. The other thing I can do with a barrier is I can have, maybe I can have a lot of positive charges on this side and a lot of negative charges on that side. What's that look like? Well, you ever seen a battery, right? And I can't remember. Is that the positive end? I think it is. And then the negative end. What do we do with batteries? We do work, right? And so the same thing with membranes. Membranes can set up these charge differentials where then I can do work across that. So maybe I can take something like sodium and pump it across the membrane by using this charge differential. But that's all a property essentially of these phospholipids, of setting up those boundaries, setting up that membrane so I can get one side from the other, and then I can use that to create potential energy, right? Which then I can get to do some work for me, right? All right, let's jump back to the textbook right quick. So that's phospholipids. That's the membrane. The membrane can have a bunch of things in it. So there's a bunch of proteins. We'll talk about them as being peripheral, so things that are just on the periphery, right? They don't go all the way through. They can be integral, or they go all the way through. Usually, or not always, but a lot of times integral proteins will be channels. So notice this one has a channel through it. So talk about channel proteins where things can move through. You can also have things like cholesterols. Cholesterols help stabilize membranes. If you have too much cholesterol, it makes them too rigid. If you don't have enough, they're too loose. So you kind of need just the right amount. You can have things like glycoproteins or glycolipids on the cell membrane. Sometimes these are used for things like cell-cell recognition. One of the kind of classic experiments in biology is, let me go to this slide. What they did is they took a sponge, a sea sponge, so we'll talk about maybe a purple sea sponge and an orange sea sponge, and they mixed them together. They actually put them in a blender and blended them up, right? And then they let them sit to see what happened. And so what happened was, after some time, you ended up with orange sponge and purple sponge. Again, they separated back out. Well, how do they know who is who? And it was through these things like glycoproteins. That's what allows your cells to know that they belong to you and they're not foreign. And so someone asked earlier, if cancer cells belong to you, why? And cancer is a product of, mitosis gone haywire. Why can't your body defend it? Well, that's one of the reasons. If the mitotic event doesn't cause an issue with something on the cell surface so that your body can recognize it as being a problem, well, then it won't recognize it as being a problem, and the cells just keep growing. Right, but that's the function of glycolipids or glycoproteins, right? A lot of receptors. Transportation across the membrane can happen a couple ways. One, it can happen through a passive process, so passive processes require no energy. Diffusion in and of itself is a passive process. A way to think about diffusion is if I have a glass of water and I put a drop of food coloring in it, right? What's going to happen? Well, the drop will stay concentrated for a certain period of time, and then it kind of slowly diffuses out. You didn't add any energy to it. You didn't do anything. It just diffused from a high concentration of dye to a low concentration of dye. And that's how diffusion happens. Another example of that is if I was to spray cologne at the front of a classroom, I would smell it first, people in the back would smell it later. That's not exactly diffusion. There's a lot of bulk flow and stuff there, but the concept is similar, right? So high concentration of cologne to low concentration of cologne. That's how diffusion works. Really, a lot of systems work that way, right? So if you think about, so we're getting ready to have maybe some interesting weather right now. And so if you think about having something like a high pressure and a low pressure, well, what is wind, right? Wind is just a force trying to equalize these pressures, right? So if I have a high pressure, right, which is kind of a downward force, and I have a low pressure, which is kind of an upward force if you're talking about weather, then the tendency is for those to want to kind of equalize each other. And so that's how you get wind. That's how you get high and low pressures, right? If you're interested in meteorology, you should study some of that stuff. It's super interesting, right? But passive processes require no energy for things to move across. I think that's talking about facilitate diffusion. Another thing, let me not get ahead of myself here a little bit. I'll go back to your text. Right. So that's diffusion, just the movement of things from high to low concentration. And it can be anything. So I've talked about cologne. I've talked about wind. It can be, we can talk about gases, so O2, CO2, right, moving across. That's how, so when you inhale oxygen, there's a relatively low amount of oxygen in the blood in my lungs, and so the oxygen wants to go from high concentration of oxygen to low concentration of oxygen. The opposite is true for CO2. In my lungs, in the blood in my lungs, there's a high concentration of CO2. So it wants to diffuse out. So it diffuses out, I exhale it out, right? And so that's why this is a terribly important concept, the idea of diffusion and transport is because it drives most every system you've got. I mean, how does nutrients get from your gut into your bloodstream? Through some diffusion process, right? So it's a really important process. You can't have things that are called facilitated diffusion. So if you know what a facilitator is, it's just somebody that helps things move along. So you can have gates and channels that allow, for instance, water to move through more easily across a membrane. Still don't require energy for it to happen. You're not using ATP. It is a diffusive process. It is a passive process. But there's just a channel there that just allows it to happen a little more easily. Right? Then we kind of have a special thing called osmosis. So osmosis is the diffusion of water across a selectively permeable membrane. Right? And so let me take a second here and maybe let me see if I can add a new. Whoops, I don't want that. I want to add a new slide. And I want to blank all this out so I can just draw. All right. So if I have, first I want to talk about the tonicity terms, right? So let's say I have a beaker, and in that beaker I have a 30% salt solution. And let's say beside that beaker I have another beaker, and my beakers are curved, and this beaker has a 10% salt solution. Right? Well, in your book, when it's talking about hypertonic and hypotonic and isotonic, those are all terms that are relative to each other, right? What do I mean by that? Well, if I say something is big, right? So if I said this stylus is big, well, big compared to what? It's not big compared to me, big compared to an ant maybe, right? So just to say this pen is big means nothing, right? You have to compare it to something. Same thing with tonicity, right? So for something to be hypertonic, right? What that means is it has a higher concentration of solutes compared to another solution. For something to be hypotonic, it means it has a relatively low concentration of solutes compared to another solution, right? And so the reason I wrote those underneath these beakers, right? So if this is beaker A and beaker B, right? Well, beaker A has a higher solute concentration than beaker B. Therefore, beaker A is hypertonic, beaker B is hypotonic, right? Because beaker B has a lower concentration of solutes compared to beaker A, right? So that's tonicity. If they are equal, if both beakers were 30%, then that would be isotonic, right? And so what does all this mean in relation to osmosis, right? Well, osmosis is the diffusion of water across a selectively permeable membrane. So if I take now, let's say I have a larger beaker, right? And let's say in that beaker, let's say I've got a membrane down the middle. And that membrane will allow water to move across and nothing else, right? And so if it allows water to move across and nothing else, let's go back and say I have a 30% salt solution and a 10% salt solution, right? And so my question might be to you, which way would water move? Would water move from A to B or from B to A? Well, the first thing you have to know is solutions are usually defined based on solute concentration, right? So if solution A is 30% sodium chloride, how much water is it? Well, it's 70% water. If a solution is 10% salt, how much water is it? Well, it's 90% water. Right? So now if my question to you is, through osmosis, which direction would water move? Would it move from A to B or B to A? Well, water would move down its concentration gradient. So it would move from a high concentration of water to a low concentration of water. So water would move in that direction. I could just as easily ask you, so let's say this membrane allowed both water and salt to move across, right? And so I would say, which way would water move, and then which way would salt move? Well, water would move from B to A, but salt would move from A to B, right? Because this is the high concentration of salt to the low concentration of salt. So things tend to move down their concentration gradients when the membrane allows, right? If the membrane doesn't allow it for a number of reasons, maybe the charge is wrong, maybe it's too large of a molecule, but if the membrane allows it to move across, it will, and it will down its concentration gradient, right? And you can even build a force through that. I need to create a blank, just a blank slide, but this will work for now. What do I mean by that? Well, by force. Sometimes we talk about osmotic pressure, right, or pressure from osmosis. And what that is let's say I had a U-shaped tube, right? So here I've got a tube, glass tube, that I've bent into a U, and then I will put a semi-permeable membrane in the middle, right? And then again, let's say that membrane only allows water to pass through. Let's say I have a 30% salt solution here. And at the same level on the other side, I've got a 10% salt solution. And then I let it go. What's going to happen? Well, still, this is 90% water over here, so it's still going to want to go down and through this membrane and up into this tube, right? And so let's make, for realism, I need to make this tube a little taller. So what's going to happen is, as water moves from this side over, right, this volume is going to fall, that volume is going to go up, right, all through osmotic pressure, because the water is going to move from this side over to this side. But it's going to push up here. What's it pushing against? Gravity, right? And so there can be an actual force created here, right? Osmotic pressure, and it'll push so high until it can't push against that gravity anymore, and then it'll stop, right? But that's osmosis, the movement of water down its concentration gradient across a selectively permeable membrane, right? Again, another pretty important concept to grasp because it has all sorts of implications later. And so what this is showing you is what would happen to your blood cells if you put them in each type of environment. So if you put your blood cells in a hypertonic, so the way to think about this is if I injected like concentrated salt water into my veins, what would happen? My red blood cells would shrivel up. All the water would leave them. You'd dehydrate them essentially. Your kidneys would have a fit too. If I put them in a hypotonic, so imagine that I injected pure water into my veins, my red blood cells would swell up. And they would swell up to the point that they would rupture because there's no, because there's no cell wall there in order to give the cell some rigidity, right? So those red blood cells would rupture. It's called hemolysis. It would be a hemolytic situation. If I keep them isotonic, or so when you go to the hospital, if you're given an IV, it's oftentimes 0.9% saline. That's considered physiological saline, or is considered isotonic. In that situation, whatever water entered the cell would also leave, right? And so it wouldn't be stagnant where no water would move at all. Some would, but whatever amount entered would also leave, and so you would maintain an isotonic state. Counter that with active transport. Active transport, the important thing there is it requires energy. It requires, and when we talk about energy in biology, we're usually talking about ATP, right? And so you can set up gradients, again, like batteries, like I said before, and that will allow some of the work to be done. And because of this, you can have pumps, right? Think about any kind of pump. Pumps are used to move things against their gradient, right? What do I mean by that? Well, let's say you want to pump water out of your pool, right? Well, if you could just open the door and the water would flow out, you wouldn't need a pump, right? What you need is something to pick the water up and move it. That requires energy. Same thing with something like the membrane, right? So notice I have a lot of sodium, right? So all these little orange things are sodium ions. So what that means is I have a high concentration of sodium here and a low concentration of sodium here. Right? So the tendency through diffusion would be the sodium really wants to go in this direction. Right? But we don't want it to go in that direction. We want it to go the other way. Right? We want the sodium to go in this direction. Right? So in order for that to happen, there has to be a pump involved. And this particular pump is called the sodium-potassium pump. Later, when we talk about muscle contractions, we'll talk about why that's important. But it's super important to maintain this high concentration of sodium on one side versus potassium. And then the only way to do that is by using something like a pump and ATP. Right? That's the important thing about active transport, is ATP is involved. Right? Your book will talk about some things like symporters, which move things. So it talks about moves two substances in the same direction. That's not as important as knowing what it means to be an active transporter. You can also bring things into the cell, so endocytosis or phagocytosis. So endo means to bring things in. Phago means cell eat, right? Phagocytosis. Penocytosis means cell drink, right? It's how cells will move water in, right? And that's all just membranes, right? Once we start to get into the cytoplasm, some other crazy stuff starts happening. And I'm not going to list all of these organelles to you because it's pretty well done here in the text and in the other video I show you. But you should read through these, and I would encourage you to maybe sit down and try to draw one of these out. So draw a cell, draw a nucleus, draw the rough ER, the smooth ER, draw the mitochondria, maybe some vacuoles, things of that nature, and just kind of look and study where they are. They won't necessarily be in reference to each other, although you should pay attention to the idea that the nuclear envelope and the ER are continuous. Right? That's not two different things. It's called the endomembrane system because they do connect to each other. So that's something to keep in mind. Right? ER, Golgi, lysosomes, mitochondria. Oftentimes here with the mitochondria, I'll get students that'll say it's powerhouse of the cell, but then they don't know what that means, right? I think it's almost a Pavlovian thing. What you really need to know is that the mitochondria makes ATP, right? That's its important function. Sure, it is the powerhouse because the ATP is a hugely important molecule for energy, but you need to know not that it's just the powerhouse. Why is it the powerhouse? And it's because it's able to convert things like glucose into energy or ATP, right? And lots of it per glucose molecule. Let's see, peroxisomes are there for detoxification. Cytoskeletal elements, there's 3 different kinds of cytoskeletal elements, and we'll talk a lot more about some of these when we talk about muscle contractions, right? But essentially you have microtubules, microfilaments, and intermediate filaments, and they're all kind of used for different things, but basically gives the cell some rigidity so that cells don't just fall apart. It also provides a way for things to move around inside the cell, almost like a monorail or a train system. It gives maybe flagella and cilia some structure so that they can do what they do. Right? That's what cytoskeletal elements does. I mean, very similar to what your skeleton does. Right? Gives you structure, gives you a means of movement. The nucleus, right? So you have the nucleolus, which tends to be a site where ribosomes are made. All inside the nucleus is DNA. You do have nuclear pores. So a common, I like to point out myths whenever I can. So a common kind of myth that was going around early on during the kind of rollout of the COVID vaccine was that because the COVID vaccine, or some of them, are mRNA vaccines, there was some idea that it would change your DNA. And so here's an example of why it can't do that, right? So this is all DNA in here, right? There is some messenger RNA that is produced, but as soon as it's produced, it leaves the nucleus and is deposited out here to be with ribosomes. It can only go in that direction. Once it's out here, it cannot come back across. Right? And so there's no way for that messenger RNA to get there outside of having maybe some reverse transcriptase enzyme or something, which highly, highly unlikely, right? But that's just an example of why that would not happen. But that's what it is inside the nucleus. Nuclear pores allow things to move out, like messenger RNA, so that you can make proteins, and that's kind of the bulk of it, really. What else? We can talk about DNA synthesis. Again, some of the other videos do a better job of this than I would, because I can't do as much animation. But what I can tell you, or what I could show you, is that if you don't pick it up here, the way DNA essentially replicates itself is, if I have one strand, right? So let's say I have an A and a T and a C and a G. The only way that these can bond to each other is A's can only bind to T's, T's can only bind to A's, C's can only bind to G's, G's can only bind to C's, right? So A's to T's, C's to G's, right? That's the only way it can happen. So if I take a piece of DNA and I unzip it down the center, I can make the other side, right? Because they can only go together in one way, right? So that's how you can have essentially one piece of DNA and then end up with two pieces relatively easily, right? And then from those two pieces, you can go on and on and on and on and on to create more and more and more copies of DNA through DNA replication. Right. A little harder concept is the idea of making proteins or gene expression. It works in a similar way, right? So if I go back maybe to the PowerPoint, it works in a similar way to replication, but it's a little bit different. So what they're showing you here is a strand of DNA, and they're telling you that this top strand is the template strand. So the idea here, so turn your brain off from replication. We're not doing replication anymore, right? What we want to do now is we want to build a protein. So what would happen is, inside your nucleus, right, you would have a template strand of DNA, right? And they're telling you here that the top strand is the template strand. So for right now, you can just kind of disregard this strand of DNA, right? Because we're telling you that the top strand is the template strand. So the first thing that's going to happen is you're going to create your messenger RNA molecule, right? And they kind of leave that M off, but that's what this is. It's a messenger RNA molecule. So inside the nucleus, you have machinery that's going to come around, and from this template strand, it's going to create a messenger RNA molecule, right? So what did we say before? T's bond with A's, right? The difference with RNA is that there are no T's in RNA. There are only U's, right? So here I have an A, so there I'm going to have a U, right? There's a G, right? Or whoops, there's a C, there's a G, right? So all of these first ones go, that one goes to that one, that one goes to that one, that one goes to that one, so on and so forth, right? And so from this template strand of DNA, you create this messenger RNA molecule. That messenger RNA molecule then leaves the nucleus through that nuclear pore that I just showed you. And then that messenger RNA molecule ends up getting sandwiched by a ribosome. Right? So you have a big piece of ribosome and a small piece of ribosome. And think about feeding that messenger RNA molecule through that ribosome like just a piece of tape. And what the ribosome will do is it'll read that mRNA 3 nucleotides at a time. Every 3 nucleotides would code for an individual amino acid. Right? So AUG codes for methionine. And I don't remember all the amino acids, but you get the abbreviations. CCG, right? CAA, so on and so forth, right? And so that all creates a string of amino acids, one linked to another, and it would give you that kind of primary structure, right? String of pearls look to a protein, so that you would end up with something that we'll talk about excision later. Something that looks like this, right? So this is a very simplified version, right? So I take this DNA, I make the RNA, messenger RNA from it. The messenger RNA leaves. This would be the ribosome, right? And then from the ribosome, you get this string of pearls, which comes out, and that's eventually going to be the protein molecule, right? And that's how you are you. That's where all your proteins come from. All we did with the COVID vaccine was say, we know that the ribosome will make a protein if it's given messenger RNA. So you then just inject and have the messenger RNA delivered here, right? So it's delivered into the cytoplasm, and then the ribosomes end up making it was the spike protein, and so your body could recognize that and mount an immune response, right? All right. Another way. So your book might talk about the central dogma of molecular biology, or they'll just mention DNA to RNA. Whoops, what am I doing? DNA to RNA to a protein, right? And that's how it goes, right? Starts with DNA, DNA makes RNA, RNA makes the protein, right? That's the central dogma to molecular biology, right? Then if we look at cell growth and division, this is something you just have to spend some time on. What you should know is that although your book spends a lot of time talking about mitosis, cells only spend a really short amount of time in mitosis, not long at all, really. And mitosis, the definition of mitosis is division of the nucleus. A lot of times students will tell me that if I say define mitosis, they'll say cellular division. That's not true. Cells that don't have a nucleus can't go through mitosis, right? So mitosis is division of the nucleus. But it's a very short amount of time here, right? It's only that. The rest of this, the cell's doing what cells do, right? They're either growing or making new DNA or whatever, right? But cells only go through mitosis in a very short period of time relative to the life of the cell. Some never do, right? Once they're fully developed cells, there are some nerve cells that would never go through mitosis. Muscle cells don't really go through mitosis, right? It's one of the reasons that muscle is so hard to heal. But you have all these gates and checkpoints here inside the cell. If you have a problem with one of these, so maybe this particular gate has a problem, right? It doesn't work anymore. That's what can lead to cancerous cells. You just get essentially uncontrolled cell growth, right? So cells grow too much. And so they start becoming abnormal. Right? Cells are also, they all come from a stem cell. And so this is just showing you, for instance, all the different types of blood cells and where they can come from. And they all originate from one stem cell. Ultimately, your whole body originated from, so egg and sperm meet, creates one cell, and that one cell gives you everything. And so it's just a hierarchy from there. Broken down as to what each individual cell type becomes. And that's one of the goals right now of science is it would be nice if we could pull some skin cells from you and turn those back into heart cells or whatever, so that maybe we wouldn't have to worry about people dying in order to give them new organs. Right? Maybe you saw the pig heart transplant that happened this past week, I believe. And so that's chapter 3. Chapter 3 is kind of involved, right? A lot of things going on with cells and cell division and DNA and making proteins. And a lot of these other, do I have it pulled up? I can do it this way. A lot of these videos that are in here for chapter 3, right, do a good job at covering those things, right? So cell cycle videos and cell structure videos. Wendy does a really good job of that. And so what I'll do is I'll upload this one into YouTube, and I'll create one for tissues for Chapter 4.